are joining online. So it's my pleasure to welcome Daniela Doain to the BNDU seminar series. Daniela did her PhD at the University of Buenos Aires, where she's from. She then moved to the neurology department at the University of Zurich, which she told me she planned just to stay for a few years and has basically never left. So she worked there as a postdoc, starting to look at the effect of traumatic brain injury on sleep disturbance. And then when she started her own group in 2017, we kind of broadened this out to look at how sleep is disturbed, particularly in Parkinson's disease and its relationship to neurodegeneration. And then most recently, and of great interest to many of us in the BNDU and in Oxford, she started to look at how you can ameliorate these sleep disturbances with closed-loop auditory stimulation. So I'll hand over to you, Daniela.
Thank you. Thank you, Andrew. I guess I have to deal with this. Right. So thank you again for this kind invitation. I could act completely nonchalant and like, yeah, every day, but I'm really, really grateful. And to be here, it's a pleasure and an honor. So yes, as Andrew said, I will talk to you a little bit about closed-loop auditory stimulation of sleep slow waves. And the painful process we went through for the implementation in rodents, because this, as you all know, started in the bedside or in the human population, and then some of the preclinical implementations that we have done so far in animal models of disease. A little overview is I will have 4 parts to my talk, a little bit of background, what brought me to the development of closed loop, the first concept, the preclinical implementation, as I said, and a little bit of the future, what we are planning for the next years to come. So what brought me to this was the concept that sleep and neurodegeneration are very much intertwined, right? This very paramount paper from Kang in 2009 showed that by modulating sleep quality and sleep amount, they modulated the amount of amyloid load in the brain of 80 animals. This is a transgenic model that we also use in our lab. And they put the animals in platforms where if they fell asleep, they fell on the water and woke up. It's a little bit harsh, but works and deprives the animals from sleep. And if they did that, this is the plot. What you can see is that the amyloid load dramatically increased. And what happened if they gave admorixant, which is an orexin antagonist, is that they decreased plaque burden dramatically as well. And what they looked at in this paper, very incipiently at that time, was that this was particularly correlated. The interstitial fluid amyloid beta that they saw was correlated directly with the minutes in non-REM sleep spent in each hour. So basically with the amount of deep sleep, which gave a hint what type of sleep was the one actually involved in this. Because with this platform over water means that you also deprive from REM sleep. But this didn't seem to be related to this. So this, of course, was picked up by the clinicians at the same time, not only in the US, but also in Europe a lot. And one seminal paper that came out in 2017 showed that spinal taps from patients showed a direct relation between sleep adequacy or sleep problems. These are different indexes that derive from the quality of sleep in these individuals, and amyloidosis or amyloid load or amyloid biomarkers in the CSF of these patients. So we were slow, but surely got there, and we started thinking, what about Parkinson's disease? So if sleep is connected with the amount of pathological proteins in the CSF, perhaps alpha-synuclein is also related to it, and perhaps then it affects Parkinson's disease. So in a first very naturalistic study, what we did was looking at a patient cohort in the clinic. It is a Zurich cohort of over 130 patients, and we looked at their natural ability to have deep sleep. So the more slow wave energy, the higher the slow wave energy, the better the sleep, right? So what we saw is that there was a direct correlation between the progression of motor symptoms in this patient cohort in a 5-year follow-up with their naturally good or bad ability to sustain deep sleep. So the more, of course, the higher the deep sleep, the slower the symptoms progression, which was, again, a big hint towards maybe there is something here between sleep and alpha-synuclein as well. We didn't know. So this is the study we did next in an animal model, in an odd animal model, some people would say. So we chose this animal model, the VMAT deficient, the VMAT2 deficient animal. Why did we choose this model? This model had been shown to have sleep disturbances. So we actually initially brought this model to our group to characterize sleep disturbances in it. It has sleep disturbances, all good, but actually what we turn up doing is trying to rescue such sleep disturbances and see whether these change the pathologies found in these animals. And what we did was first a pilot, a dose response curve, basically, where we looked at the changes in the delta frequency upon different concentrations of this drug. This is something people have never done before, which is very surprising considering this drug is used in patients already. But nobody knew exactly the dose response. So we aimed at something specific here. So we didn't want to do what Kang did, which was modulating the amount of sleep and the amount of slow wave activity, but we wanted to only modulate the amount of slow wave activity without changing the total amount of sleep. And that's why we selected the 200 milligram per kilogram for the next experiments. So this was an acute experiment, and then we moved into the longer experiment within the study, which we did for 4 months. So what we did for these 4 months, right in the brink of synucleinopathy, when this model is described to start the accumulation of alpha-synuclein in the brain, which is at 14 months. So we started at 14 months and we finished at 18 months of age. And what we did was a twice daily oral administration of 200 milligram per kilogram sodium oxybate at 9 a.m. and at 5 p.m. And then we had a control group that was administered with placebo, and we had a sleep deprivation group that actually received the platform over water. So there we could not be that specific. We went back to the method introduced by Kang, and we used this to have a counterpart, a negative effect over sleep to see what the effect was in alpha-synucleinopathy. So in a nutshell, this is what we got. So I put here a generic plot. This plot is not in the paper actually, but this is one of the ones I like the most, where you can see a control animal, an animal that is transgenic for the mutation that increases alpha-synuclein in the brain, unclear how, but it does. An animal that was sleep-induced and an animal that was sleep-deprived. And you can see when we quantify this, particularly here, I put the plot of phospho-alpha-synuclein, so phosphorylated in the serine 129, what we see is that the placebo is different to the placebo control, which means the animals were sick in the first place and accumulated synuclein in this period range, which is important as a positive control. And what we see is that when we sleep enhance the animals, there is almost a decrease towards a wild type level. So of course, this level is not zero. These animals are 18 months of age. So there is a natural accumulation of alpha-synuclein in those brains. And what happens when we sleep deprive these animals, I didn't say this, we deprive them for 16 hours a day. So they were 16 hours a day on the platforms, and then they had a bit of a rebound. And what happened is that we dramatically increased the load of alpha-synuclein. You can see it here. The number of cells that we could count that were loaded with pathological alpha-synuclein was much bigger. And when we confirmed this in Western blot, what we saw is that the sleep-deprived animals were the ones accumulating the most proteinase K resistant alpha-synuclein, so aggregated alpha-synuclein. All good until there. So we saw something similar in different terms and by a different approach that what Kang saw in the Alzheimer's mice, but they never explored how. or they did in subsequent papers, but not in that paper. And they pointed always to a role of protein production and release. So neuronal firing that is different between different sleep states will modulate the amount of production and release of a certain protein into the extracellular space. That's their hypothesis. Then there is this other hypothesis about proteins being actually regulated in the extracellular space by lymphatic clearance. So lymphatic clearance is supposed to work to be boosted during sleep, to have a more efficient function during sleep, and thus clear proteins from the extracellular space, potentially making less available to be uptaken by another healthy cell and protein spreading in the brain. Those two options were great, but we decided not to stay stuck. We tested them in separate experiments, but we decided to go more with the fishing nets. I said, well, this cannot be one process, another process. They are not also mutually exclusive. So what about looking at the proteome of the brain after sleep modulation? So this is what we did. We extracted protein from these prefrontal cortical areas. These animals strangely accumulate synuclein in the prefrontal cortical area of the brains. We had a big group of each, so 6 animals, at least 5 to 6 animals in each group, and then we did a shotgun mass spectrometry. This I did in collaboration. Don't ask me the technical details, but what we did is we identified over 900 proteins that were significantly modulated. So clearly not a couple of pathways, a lot of pathways. When we subjected this to analysis, by functional clustering, what we found is that there were at least 150 clusters that were modulated significantly, these proteins corresponding to each of them. And because we were very, very lazy, we selected only 10 of such 150 clusters and looked at them in relation to the different groups and in the relation to neurodegeneration. You can see here a list of these 10 clusters, their enrichment score and the number of proteins in each cluster. I'm sure here we only untapped the tip of the iceberg, but we have protein folding response, lysosome function, proteasome function, oxidative stress, exocytosis, ubiquitin function. So obviously all processes, all functional clusters related to protein homeostasis, to cellular homeostasis. And you can see how they were modulated here. So take this animal, it's a wild type placebo, right? This is the expression level in this animal. If you compare with this animal, you can see these proteins, these pro-homeostatic, pro-proteostasis proteins are all reduced in the transgenic animals, which may already explain why they were accumulating synuclein in the first place. What happens when you enhance sleep? So this is slow wave enhancement, right? With sodium oxybate. You recover, at least partially, the level of expression that they had in the wild type animal. And what happens when you apply sleep deprivation for these 4 months, it's a very harsh protocol of sleep deprivation, is that you completely blunt the expression of these proteins. There is barely any expression in the sleep deprived group. And that is how protein accumulation rises up incredibly. I leave this as a side note. You can see the wild type animals don't like that much sleep enhancement, which is a kind of mental note to be very careful what one does in a healthy individual, in a healthy level, right? You would think, oh, they will benefit anyway. There was nothing to do. So they did not decrease their expression as much as the sleep deprived, but they decreased it to a significant extent compared to the untreated animal. So perhaps sleep enhancement in a healthy system is not the best idea. So basically, this is my background, right? So until here, we say, well, there is a well-established connection between protein homeostasis, sleep disturbances, perhaps other functions such as inflammation, glial activation. There are many, many other processes that we are looking at the moment. And sleep disturbances and the accumulation of protein. So there is a little bit of a cycle here. Question is, can we keep on attempting to give pharmacological modulation to modulate this cycle? Are we specific enough giving a drug that modulates or enhances low wave activity to do this? And the answer, my answer nowadays officially is no. Actually, the answer of the clinicians was not. I had been promised the promised land, and I had been said, if we work with a drug that is approved for the use in patients, we are going to move into clinical trials. That did not happen. So this is their arguments, and today my arguments. There is a lot of tolerance, there is a lot of abuse, difficult personalization, what is the dosage for each patient, misuse, a lot of misuse, particularly with this drug, side effects, dependency, and you name it. And particularly is important in our conditions as basic researcher is the specificity. So this was a GABA B agonist. It did a lot of things, not only in the brain, but also in the body. So, all right, something more specific, let's go for it. What was more specific and what was happening at the time all of this, we were cooking all of these ideas? And Go came out with this brilliant paper in 2013, showing that auditory modulation targeted to the up phase of the slow waves had a positive effect on power in the delta frequency range. And that this ameliorated retention in a memory task. Later on, Reto Huber picked up this idea with the work of Sarah Fattinger, and he did this, but instead of targeting the up phase, he targeted the down phase of the slow waves. And he showed a negative effect on recovery and memory retention. So there was something non-invasive, quite non-invasive and highly specific coming out. And then I decided, well, this is my shot. I will give it a try in the rodents. And this was the concept. This was our first drawing. And I hired a bioengineer, very naive, very intelligent, and a great person that trusted me enough to build this from absolute scratch. And this is what he did. I was telling Andrew yesterday a little bit about the story, how we build these boxes. No such boxes existed. So he literally cut wood and plastered insulation material and build the lights. And of course, the friends at TDT for a reasonable price provided the modulation system that basically completed our setup. So these animals are in these auditory insulated chambers with their own lights. We can have different rhythms in different animals. They have a loudspeaker, they have a mobile swivel. Obviously, this is tethered recordings, but with a very light cable. And we can do here rats and mice. So the bottle would cover the rat. So the bottle goes here, they have a normal environment. So the first thing we did was diagnostics. Does this work? Is precise? Is it specific? Is it sensible? So what we saw is that we could get a high level of specificity. We were very, very precise, but we were not very sensible. So we only stimulated some of the slow waves, right? And when they asked me, I say, well, this is the gold standard, just because as I was the first, I say this is the gold standard. But actually, today we know that we can do a little bit better than this. Then what we started doing is delivering the up phase, down phase, and a mock, just flagging events to see whether we could track these slow waves well enough, stage them in real time well enough to be precise in our targeting. And this is what we saw. So we were really, really precise in our flagging and in our delivery of stimulations to the 60 degrees of the up phase or the down phase at 270. So, right, did it work or not? So we were targeting, we were doing it right, but did it work? So we went and looked on specific days of our protocol whether we had an effect in the delta frequency band, and we did. So this is an individual day, but I will show you now for a prolonged period. So we have a decrease in delta power upon slow down phase stimulation, an increase. It's a little bit more than the delta, I would say. It's a bit extended, but the main effect is here and nothing in the mock, obviously. And this is what happened in a 14-day protocol or 16 days protocol, I think, with the behavior. So we can see that the effect is constant, and it's not only throughout the day, but constant between different days, right? The mock is a little bit of a mess. Everything happens depending on the behavior and food anticipation. And in the down phase, you can see a very stable, steady decrease throughout the day, which is very interesting. So almost no rebounds. There is a little bit something here in the first days of on the last days of the protocol, but nothing overall significant. So what happened with sigma, right? Because this is a little bit the theory behind the memory model, right? If you are driving slow waves, you may be driving spindles as well. So we looked at sigma activity. We also had an increase, and it was specific to time in certain parts. So we had a main decrease, for the down phase in the first hour, so in the light period of the day, while the increase with the up phase was kind of throughout the day. And this is non-training sigma, because I say this because there was a sigma effect promoted only by the behavioral training, so we need to subtract that. So this is already the normalized increase or change from baseline, and we can see a significant increase upon up phase and a significant decrease upon down phase. So we are driving in a way or depressing the amount of spindles upon slow wave activity modulation. So this has to have an effect on memory. This is what we thought. And we did, not a memory here, we did a learning task. So we looked at how the animals learn over a period of time, how to grasp pellets. You all know this, right? Through this very narrow window, they have to learn to put their paw through it and grasp a small pellet that is sitting on a pedestal. And this requires a fair amount of training and learning until they are very good, right? So we did this in a pre-training and then motor training and non-training days. And this is what we found. It's okay. It's not fantastic. So we could not actually corroborate the showings of Ngo that he improved memory with upsides. We couldn't see that. This might be related to species. This might be related to our small effect size in the increase of slow wave activity. We were between 5 and 15%, so it's not so big. But down phase instead had a positive effect. I mean, a significant negative effect on memory. So not only they decreased the amount of attempts and the total attempts over time, but also their performance was significantly worse than the untreated animals. So we corroborated instead what Reto Huber had done with the down phase stimulation. So we have a negative effect on memory while we are decreasing slow wave activity. So we said, well, we are good enough. Memory is anyway not our main priority. We wanted to look at the pathology. So we said, this system is specific enough. We have good, stable, solid effects on slow wave activity. Let's go look at the next. So no, this is only, I forgot, correlations on how actually the changes in delta and in sigma drive the success rate. So there is really a correlation between our increase and our modulations at the EEG level and the memory outcome. But then, as I said, we wanted to look at this in the pathology model. But what pathology model? At that time, I was kind of still hung up with this traumatic brain injury model that Andy mentioned I was working with and I developed during my postdoc years. So I decided to look at that, just because I had good markers to look at pathology, because I had good behavioral outcomes to look at. So this model, it's a model in which a rat is sit on a little foam platform and receives a blunt hit over the scalp, which is protected with a metal plate, and develops diffuse axonal injury. So accumulation of proteins in the axons in different brain areas, cortex, hippocampus, cerebellum, corpus callosum, and so on. And we also see that there is a progressive decrease of recognition index in the novel object recognition test. So we have that model. Is that this was an earlier paper. So we decided to use now our down phase stimulation over this model to see whether we could counteract the effect of sodium oxybate. So we knew in this model that sodium oxybate had a positive effect. And we knew that shorter periods of sleep restriction had also led to a sleep rebound that had also a positive effect. So can we counteract with down phase the effect of these two classical methods of increasing slow wave activity and thus reverse their positive effect? It was just our way to comparing the strengths between a classical method to modulate slow wave activity and acoustic stimulation. So we did the regular checks. We looked at non-REM in minutes in the sham and in the TBI animals. Nothing was changed. Oh, good. This is what we always want, to modulate slow wave activity without changing sleep amount. And then we looked at the effect of mock class, so not doing anything, or down class in the different groups, in a placebo group, so basically a standalone down class, in combination with the sleep restriction group, or in combination with the sodium oxybate group, just to see how was the relative strength of the methods. So when we apply it standalone, what we see is that there is a positive effect of a significant effect of down phase stimulation on slow wave activity. So we decrease delta activity significantly. We already knew, but this is not trivial. We didn't know if we could do it in an animal model of the sleep. So we had done it before in healthy animals. So that checked out. That was good news. What was not so good news is that down class could not counteract the enhanced slow wave activity elicited by a short period of sleep restriction, or even so less. Here there is a little bit of variability that could suggest maybe if it was a little bit stronger, we could have, but here there was not a dent, not a dent. So when you give sodium oxybate and you enhance slow wave activity, there is no way you can bring it down with down phase class. And of course, then, as I said, when we give this sodium oxybate or this sleep restriction, we have a 90% reduction on diffuse axonal injury. This is a story that was published earlier. So can we counteract it? We don't think so, but let's take a look. And so we did. And what we found is that what we count is these axonal bulges, right? This is just an example of what we look at in the microscope and do stereology on. And what we saw is something interesting. So down phase stimulation didn't make it worse per se. So it did not make the pathology more severe in these animals, but it also did not rescue the rescue. So the rescue that we exerted with sodium oxybate, you can see the level here is around 6,000, and here it's about 3,000. So this is a rescue that is significant. We know it to be significant. We could not counteract it with down class. Meaning the take-home message of this paper was, it works in a disease model, but it's not that strong. So one has to be careful on the conditions that it's applied on. But what about up phase? Because that's the key question. Who would want to give an animal model of disease even worse sleep. No, let's give it better sleep. So this is what the rats look like. This is just a video. It's stills from a video that we need to do for the journal. And so when we did up-phase class, did it work? It did. You can see here, this is a baseline day. This is 5 days of stimulation. Why do we do it 5 days? It's because this is the period window where traumatic brain injury is sensible to something. After that, everything is a debacle and there is not much room for improvement. So we did these 5 days like we did before with pharmacological modulation, and we can see an increase in slow wave activity that doesn't go back to baseline. And this is just because traumatic brain injury animals, you can see it here in the TBI mock class, they have an increase. It's transient. We have studied it before. It's transient. It goes from day 5 to day 8, and then it just renormalizes. We don't know what that's about, but this is basically our new baseline level in the control, and we can see we have an increase, and this goes back to the new baseline level, right? While the sham animals just remain around the 0. And we did this without changing fragmentation and without changing sleep amount. So our controls still stick up. So what happened with this modulation? Can we rescue the pathology? It seems that we can. I'm very hopeful. I still don't know how we do it. I'm still studying clear activation and other pathways that may be involved in this. But actually, we have a strong reduction after up-phase class in traumatic brain injury, in the axonal valves in traumatic brain injury brains. And we almost go back to healthy levels, to non-TBI animals. And this is significant. And what we see is that in the TBI group, this drives a correlation between the amount of slow wave activity change from baseline and the number of valves that we find. So the animals that have the biggest increase are the ones that had the least amount of valves. But then, is it only protein? So are we really doing something that protects the brain, or are we just clearing protein? Because we know sleep may have a direct effect on protein clearance. So are we really affecting processes that will change the disease? So what we did look at now is something different, which is a structural marker. We look at myelin basic protein here. So in the corpus callosum, particularly in this area, which is particularly affected by the heat that the animals receive, you can see a decoloration. I don't know if you can appreciate it. So in this particular area, you have less intense staining of MVP, which is the most abundant protein in the myelin sheath. So basically, these axons are demyelinating after traumatic brain injury. And what happens after up class was really surprising to us. It's the first time we see an integrity marker, a brain health marker that is actually changed after closed loop auditory stimulation. It's not such a dramatic effect, but it was not to start with. It's a very mild model that we do, so we have a significant effect. And we recovered this to non-TBI levels, so to sham levels. And you can appreciate it here in the picture. So it's a much more intense staining here. And again, this correlates with the amount of slow wave activity. So those animals that had the highest change from baseline, that responded better to the stimulation, were the ones that had the best staining quality. So the most recovered myelination, or preserved myelination. I don't think the demyelination started. I think what we managed to do is arrest it before it did. But yeah, right, all good. Patients won't care if I tell them, yeah, the demyelination is good, but you are still messed up with your memory. So what we did was testing memory. And what we see is that we have a significantly different from chance level recognition index in up class. This is a little bit of a weaker experiment because many animals need to be excluded from here from stress reasons and pain reasons. but still the remaining animals, we needed to do a median split, and we kept the animals that actually responded well in the test from all of the groups. And the median split animals are not that many left, but still we can see a significant preservation of memory in the novel object recognition test. So kind of closing our story. And I will speed up to have time to tell you about the latest things we have been doing. So all of this was to test whether we could develop class, we could use it in disease models, but my real interest is to apply it in models of neurodegeneration. So then is when Ines joined my group. Ines, it's a senior PhD student that will be looking for postdocs next year. And she developed this in the mouse model, in different transgenic models. So we had 2 baseline days and 2 days class, and we tested many different conditions. But then we have to hit the brakes just a little bit, because actually we realized kind of early in the project that we could not apply the rat parameters to the mouse as stimulation. And the reason for that is that between rats and humans, there is kind of a similar pattern in the peak of slow wave activity, right? It's a bit similar. It's not exactly the same, but it's a bit similar. But this is how it looks like in mice. So it's terribly different. We looked at 0.8 hertz peak frequency for humans, 1.35, and 2.5 for mice. So we needed to really adapt not only the stimulus duration that we were using before, but also the frequency being tracked in real time to stage slow wave activity for it to be efficient. So we started with a fixed phase target, the same one that we use in the rat, at 60 degrees, and with, say, different frequencies. This is all of the transgenic animals, mixing AD and PD animals here, and all of the wild type animals. And we tested different frequencies being tracked. We looked at 1, 1.5, 2, 2.5. And then we decided the best increase was, the best precision as a diagnostic was given by 2 hertz. So we fixed this one. And then we moved on to having that a fixed variable targeting different phases of the slow wave, because 60 maybe was good for the rats that had a slower wave, but maybe we had to kind of advance our target stimulus. And so we did 3 different phases, 30, 40, and still again 60 to see. And a long story short, what we saw is that 30 is the best target for AD mice and for wild type from the AD mice, and 40 is the right target for transgenics and wild types from the PD line, which was rather surprising. So I don't know, with some of you, I discussed this yesterday. What we realized is that the overlap between transgenic and wild types from the same strain is almost complete, which is not necessarily expected. We expected the disease to have an effect on how the waves characteristics are and how we can track them. But this was not true. Actually, the biggest difference was between different genetic strains. So it's a bit of a cautionary tale. Be careful when you select your strain if you are going to apply a closed loop, because it might be the case that you need to completely tailor your parameters. But yeah, we had a reasonably good phase targeting. You can see very interestingly, if we would have kept the 60 target, we were falling off of the up phase already, with majority of our triggers are out of the sweet window, which is here labeled in gray. So this was too slow. This was too late a target, and it was already falling outside of our interest zone. But yet, so what did we get? Did we increase? Yes, we increased. And if you look at the numbers, we are close to, depending on the groups, 30 to 35% increases in slow wave activity compared to the baseline, both in wild type and in transgenic animals. We have very, very strong, stable effect of auditory stimulation with the up phase here. But then Ines decided to give it a first test run. So how does it look like in terms of acute effects onto sleep characteristics in the different models. So she looked at non-REM. There were big differences known to be there already in the AD line. She did not change that. So we still stick with the fact that auditory stimulation does not change the total amount of scored non-REM sleep. And same happened, so when there was no disturbance, also there was no rescue. But what happens with fragmentation? In the Parkinson animals, you see there is no disturbance to start with at baseline, so there is no change. There is no rescue there to happen. So it does not mess up with the equilibrium in this model. On the contrary, what happened is that there was a big difference in fragmentation at baseline, with the transgenic animals being much worse, having much more fragmented sleep. And what she showed here is that you can decrease it with up-phase class. You suppress the transitions. We still don't know how, but we are looking into it. So when there was a disturbance, we could ameliorate it. And this is based on EEG signal, right? So we look at EEG signal. And then she asked herself, well, can we also look at another way to look at fragmentation that is not based on EEG? Because EEG is we are looping into the EEG and looking into the EEG. So perhaps we need to look something that tells us about the behavior of the animal. So she looked at microarousals based on EMG power. So based on the muscle activity. And what she saw is that the transgenic animals had a baseline pathological level of microarousal. So there is a lot of muscle tone in many different, particularly in the light period, in many different instances, right? And this is severely increased at baseline. And what happens is that you almost blunt it completely after stimulation. So there is a relaxation of the muscles effect that we are studying further now. But another way to say we had an effect on fragmentation. And this is the very last thing. This is completely unpublished. This is new stuff. We are working now on the long-term effect of auditory stimulation in terms of behavior, in terms of brain pathology, in terms of biochemical outcomes, in terms of blood biomarkers, and more recently also we started looking at the retina as a first initial window. What is happening with the pathology upon 2 weeks of up-phase class, so longer-term stimulations? This is really, really out of the oven. So this is the effect of acoustic stimulation up-phase in the retina. So we see a lower accumulation of amyloid precursor protein in the AD animals, and we see a decrease also in nitrated synuclein. No, sorry, this is oxidized synuclein, synuclein 303, oxidized synuclein. So another form of pathological alpha-synuclein accumulation in the retina upon 2 weeks compared to MOC. So first preliminary data showing that the central nervous system here, mirrored by the retina, has benefits from up-phase class stimulation when it's done in a prolonged period. And the very last result, so we looked at the behavior. This is also very preliminary data. We are still completing the groups, and we calculate success rate, latency, and performance. And what we see is that the animals got, so the animals, this is AD animals, the animals got better in success, not significantly, there's a trend, but we think if we add more animals, we are still completing the groups. This may check out, but also became slower doing their decision. So when we compute this into performance, what we find is that there is a significant rescue of the decline that the placebo animals, the MOC animals going through just flagging and no stimulation have over the 2 weeks period. So you can see performance compared to baseline decreases over time. This is presumably the disease process taking place. And in these ones, there is no change compared to a wild type animal. So first preliminary result that we might have an effect on memory as well. So done. I hope I did not go too much over time. I just want to tell you where we are today, where we think we are today. So is class in rodents is feasible, it's safe and stable over prolonged periods. It requires model-tailored parameters to work. Genetic background may be one of the biggest factors there. Slow wave enhancement ranges between 5 that we've seen in the rat and 35% that we see in the mouse. Is this species related, or is this we just were too lazy with the rats that we did not do this very, very specific targeting, or this very this very good optimization that we did later? We don't know, but whoever is going to start doing class, consider this a possibility that you need to really work with parameters before. It modulates sigma power as well, spindles potentially as a proxy of spindles. We have not yet counted spindles per se. We are starting to do that, and learning performance. It has acute effects onto neurodegeneration-associated sleep alterations, fragmentation particularly, but not only. I couldn't show everything. And longer-term effects over central nervous system pathology based on the preliminary retina data, and also on symptoms, the memory in the AD animals. So where we are going from now is, of course, we have a lot of questions. What are we doing? What are we doing? In terms of circuitry and cellular mechanism, what is the pattern of neuronal activity in deeper brain areas, in the thalamus, in the auditory cortex? How is the circuitry playing a role? Are these slow waves generated in the thalamus? Are we driving them from there, or is it just a cortical effect? So we are now implanting multi-arrays. Then we need to understand how the proteostasis works. So another project that is starting in the lab is brain microdialysis in real time with closed loop auditory stimulation. So we need to see what is the protein release and clearance in real time, because it's very easy to sacrifice animals at the end and look at the brains, but we don't know what is the temporal dynamics of these associations. And not only clearance, but also other outcomes like autophagy and microglia activation. And last but not least, we need to look at performance and recovery more. So we need to look at how the transitions between different states and the potential suppression of these transitions makes sleeps more efficient, and potentially the change in the architecture of slow waves as well, not only wavelengths, but other parameters. We are studying their association with performance, and we need to look not only at microstructural, but microstructural changes. So I could thank a lot of people for this, but I will thank these two individuals, Carlos Moreira, who developed the rat closed loop auditory stimulation from scratch, and Ines, who followed up and was brave enough to transfer it to mice. They are both extremely talented and looking for positions. And of course, our funders and our animals. Thank you.
Thanks a lot, Daniela. It's an amazing body of work. Any questions?
I've got a couple. First is, have you tried to look at the effects of different phases around the up phase or around the down phase and the effects, or was the 90 or minus 90 degrees the most effective?
You mean onto the outcome measures?
Yes.
We might have some preliminary data, right? So I think Ines has behavioral data and some histopathology done on the different phases, but we stick with the one that gives the highest precision and the highest increase in slow wave activity. But it's not a bad idea to look at the dose response in a way. So how the targeting affects slow wave activity in different amounts and how this affects the outcome measures. We should do.
Lucas?
Yeah, absolutely fascinating.
Mhm.
I have a question. Why do you think you don't change sleep? Because I think in human class, Elena Kugler from Rita Huber's lab has shown basically when she boosts slow wave activity over the first half of the night. In the second half of the night, sleep is more superficial, suggesting that sort of by making sleep more efficient, then afterwards sleep is sort of more superficial. More
Right, but she did not see a total change in number, I think, either. So she thought the distribution of deep sleep has changed. So when she started, so it's biggest in the first half of the night, but I don't think she's seen a change in total number either. So I'd argue we changed the transitions. I think our money is now on the transitions. So we might change. I will not anymore say we don't change macrostructure on sleep, because that's what I used to say. I think we change the transitions and we affect fragmentation, but not to an extent that will change the scoring. So we are talking about scoring, right? If we think of sleep as fluid, then we might have a different outcome. But when we do the 4-second epoch scoring, we do not see a change, a quantitative change in scored non-REM. And I don't think Elena does either.
Maybe just a quick follow-up. Have you tried, for example, stimulating for 4 or 6 hours and then checking if afterwards sleep is deeper or more superficial when you disrupt or enhance?
Right. No, we have not done it. So all of our protocols have gone continuous 24-hour recordings and stimulations, so either on and off. What I can tell you is that Carlos, this is a result I didn't show. This was asked by the reviewers. So he sees that there is a complete renormalization. So he has an increase in slow waves from baseline and then switch off next day, 24 hours, complete recovery. So we don't see a rebound even in sleep amount or in slow wave activity, which is surprising.
Please.
Thanks, Daniela. That was super. So age is one of the biggest risk factors in AD and PD. The biggest if you don't have a genetic underpinning for it. And my understanding, perhaps from Penny Lewis, is that the older people get, the smaller their slow oscillations get,
Right.
the lower the power. So you're starting perhaps to lose your signal with age, your triggering signal. Do you think that's a confound that can be overcome with the rat studies, mouse studies? Is that an issue? Is everything quite young here?
No. It's younger than in pharmacological studies, so we don't age the animals until 14 months anymore. We change models so that we can work in an earlier phase and still have the same amount of synucleinopathy. But they are 7 months, so not that young, I would say, for a mouse.
That's sprightly middle age. That's fine.
Exactly.
But we have this phenomenon in people,
Right.
right? And I would imagine in AD and PD, their slow oscillations are small.
Right.
So that sounds like a little bit of a confound for ready translation. Would you think that can be overcome through different signal processing or another way of capturing?
Right. No, I agree with you. We have a paper published a couple of years ago on how different ages in the AD model that we use, how different ages compared to age-matched wild-type animals. And what we see is that the wild-type animals lose their slow wave activity capacity at a certain age, and the AD animals have exactly the same process but happening earlier. So this potentially should be something we compensate, but because we always compare to the wild-type animals that are age-matched in every single experiment from the same litter. So we feel like we are kind of normalizing to those. However, the level is at that age, we are comparing to that. But I agree, it is worth to look into.
One of the important controls, because you said, if I understand correctly, you only hit about 10% of oscillatory cycles. So in any comparison of animals, you'd have to say, show the total number of cycles you hit.
Right.
Is the same in this group?
The density of trigger.
Yeah.
I agree.
Do you see that?
I agree. We have a bit of a difference there in the transgenic animals. We do.
Okay, fine. Thank you. Thanks. We'll discuss.
Right.
Do you think there's any evolutionary adaptive value to having auditory stimuli affecting slow wave activity? And why does this happen in the first place?
So there is this whole concept of gating, right? So one, in theory, environmental noises should keep you on a higher alert and evolutionary, right?
But why would it be then so dependent on what
Right.
happens on the?
Right. So that's exactly what we want to find out. What is the processing? Is it going through is it purely acoustic stimulation altering the auditory pathway in the brain, or is there other mechanisms at play, right? Hippocampal recordings could be interesting. Amygdala recordings could be interesting, right? To see whether this is in a way gating arousal in other areas upon down phase or upon up phase. I don't have an exact answer. How we think it today is that if you are if you are in a swing and you push the swing in the right moment, you kind of boost that activity, right? What is the evolutionary value of it? I do not know. I could guess half an hour, but I don't know the answer.
I had two small questions, but they are linked to what Pete and Dave were asking. The first one is, in your work, which is massive, okay, you have a lot of models. You have where you have two species here, and then you have PD, AD, TBI. I was just wondering first, if you were to apply class to aging, naturally occurring aging, would you somehow support or prevent MCI?
That's a good question. We are discussing this, yeah.
Because there's a lot going on, and the sleep deprivation is highly stressful. I mean, if I was to sit on a platform, to not follow water for hours and hours, I would just be stressed.
Right. So we control for stress there in that study.
Like
right.
Naturally occurring aging, like sleep architecture is changing as a function of age.
Right. Right. No, I agree. Those are two separate questions. So about the platform, you are right. So we control therefore body temperature, we control for food intake and for body weight. So we had course supporting that also because the veterinary office was on our back with that protocol. So we needed to show there was no signs of stress. We did not measure cortisol, but can measure cortisol, and he did not find change.
Commenting on what's more, I'm just curious about whether you think this class could be applied to MCI and then.
Right. Yeah. So this is a collaboration that we are discussing with Mark Sust. So he is looking at the uncoupling of spindles and slow waves across the lifespan, right, in healthy individuals and potentially in MCI patients. And amyloid pathology related to this uncoupling. And this is something I want to look at in my models now in relation to class stimulation, whether we can suppress the uncoupling or restore the healthy coupling related to the age window and see whether we can change pathology outcomes and whether we can prevent memory decline in the age range. But it's a project that will come.
My second question related to Dave now is I used to have this old clock during tick night, and I just can't sleep, so I just switch it off. Okay, but if I have an ongoing signal like a white noise, I'm fine. I was just wondering whether you are sending discrete pulses right back to the animal. It's a tick.
It's a pink noise.
Have you tried another modulation of your slow waves?
No, we haven't. Caroline Lustenberger did it in healthy elderly, in patients with binaural tones, and she looked at a lot of it. And if they are sub-threshold, so if you work the intensity right, they don't wake up more. They do not wake up more. So this is also something I didn't mention. So Carlos worked on a huge pilot before really starting the stimulation on stimulus duration, stimulus intensity, of course, some phase targeting, and we really needed to be on the lower intensity levels for this to work. So I don't think they consciously hear it, the brain is processing it in the background. So So I would not see a parallel directly with your clock, because your clock, you're hearing it.
So it was more meant like if you wanted to modulate slow wave sleep, what about just playing an analog modulation? So you on the fly, you keep track of the phase, but then you enhance or you do an anti-phase, but the signal you are going to send is analog, if you know.
What is exactly the question? So where?
So in the class, what you send back to the
Right.
memory is a click.
Yes, it's, yeah.
Instead of on the base, you put it on the
Ah, I see.
continuous.
Right.
stimulation.
So-
have it as-
He did not do a continuous, but Sven Lea from the Reto Hoover group, he did down phase and up phase modulation of K complexes. And he sees a positive effect on memory on both, on down and up phase. So there is something to it, right? Depending on the type of waves that you are stimulating and reading out as your outcome measure, there might be a difference. So what we are targeting here, given the thresholds that we set in the system, are type 1. We are looking then at type 1 and type 2 combined, presumably. This is what I'm trying to figure out with Miguel Navarrete. So we are trying to understand a little bit more what type of waves we are really targeting and in the different hours of the 24-hour period. But it's right what you said. So Sven is stimulating K complexes, which type 1, right? So he sees no change from up phase to down phase in memory.
But one online from Andy Jackson,
Right.
he says, does he understand correctly from your early slides that you're running the class protocol 24 hours a day?
Right.
And what happens if you restrict to sleep or wake periods?
Right. To sleep or wake periods or to dark and light phases? Because
He
we
says wake versus sleep.
well,
I
we
guess that's
restrict,
also-
we are staging sleep in real time, right? So
Yeah.
this only have this 10%, 10-14% in the case of Ines is only happening during non-REM or majoritarily happening during
As
non.
in switch the closed loop on only.
As it only, of course, we have a real-time staging
Right.
that detects slow waves, and when this happens, then the algorithm is complex. So once when we are in slow wave sleep, then the thresholds start activating in terms of beta and delta ratios and EMG. So yeah, but this is in the Carlos paper, very well explained. So we are restricting to non-REM stimulation. We are not awfully precise. Of course, there is off-target modulations, but yeah. And if we have to do it in the light or dark period, well, I would argue that in the plots, in this heat plots, heat maps, we see an overall increase throughout the day. So I would not see a reason. It's potentially a basic research question of interest, but for pathological modulation, I want the best effect, the most robust effect possible. So any non-REM event that likes to be targeted and is picked up by the system goes for now.
Yeah, couple of final ones then. Yeah.
How quickly do you see effect on the activity we can turn on stimulation? We need to take a period of time to build up the effect.
Good question. I mean, of course, as soon as a slow wave event is detected by the algorithm, it starts being stimulated. So in terms of stimulus delivery, it's immediate, right? But the effects on slow wave activity, I think I have a plot here. This one. Well, this is on wavelength. I think it's pretty immediate too. If you look at the hourly value, I don't have the plot here to show you, but I can email you. So if you look at the hourly values of increased slow wave activity, you can see already an increase in the first 2 hours, already, depending on the bout of time that you select.
first one is, everything we look at the so the effect is, then within the extent, again, with the highest efficacy, it could be increased and actually rate, really, the maximum 40 but when you have a high degree of recommendation and or because we use non-rem efforts and one for instance. So have we looked at the effects of the CT targeted sleep states, the effects? And the second question is, and I will test because I was the TPR more. I presume this is now more for TPR.
Yeah, Superman.
So the first question that I had, I wonder if we can think about this one, have you seen it?
Right. So Ines is starting to do that exactly in this analysis. So she's looking at the transitions. So this is state space analysis where each point is about of a certain vigilance states and so we have very preliminary data that I did not want to include, but she's looking at how these clusters of wake, non-REM and REM are linked or separated, and you can calculate the distance from the centroids and the velocities of transitions. And this is known to be effective, for instance, in Parkinson's disease. They call it bradysomnia. So the preceding state determines the velocity of transition to the next state. So she's starting to look at this type of analysis. So where is it coming from and how it's where it's going, the velocity of it, the distance between these transitions, and whether class affects this or not in the disease models. But I have no solid data. Sure.
Hey, I think we better end it there. If you'll join me in thanking Daniela again for a great talk.